So I'm just going to show you some movies and animations of what this looks like, and we'll come back and focus just to summarize some of the things that we've talked about so far. So this is actually something that people have been looking at for a long time. This is a beautiful diagram of drawing of a cell, actually. Here you see the chromosomes and these dark shades, and these two pointy ends here, that is the spindle again. This is actually an image that was drawn more than 130 years ago by Walther Fleming, who was just looking down the microscope and saw this and drew it by hand. He did a remarkably good job. And I think that not only did he see these things, he could do it by time lapse. So here's another good drawing of what he achieved. Again, these sausage-like things are chromosomes. And so I'm going to show you actually the world's oldest time-lapse movie of mitosis. So here he was just drawing this periodically every few minutes. Come in, you can see these sausage-like shapes, and you can see the chromosomes sort of move around. This is prometaphase now. Now they're aligning in mitosis, metaphase. Now they're distributing, anaphase. And then we'll draw in a second here this final phase where we've got these two new cells, and they're going to break off into two different pieces. That's cytokinesis or telophase. And now he has two new cells, right? And so you can actually see this process happening. This is what this looks like in slightly more modern microscopy. So here's a movie that was taken in Ted Salmon's lab at UNC. The sausages are chromosomes again. And you can see these sausages when the movie begins, and it will cycle through. When it begins, they're sort of spread all around the nucleus. And so we're going to watch it play again here. So the chromosomes are there. The nuclear envelope breaks down. They're released. They start holding onto the microtubules. They move in the middle. And it's not actually until each of the chromosomes is aligned in the middle that now they're going to all synchronously just go bam and separate away from each other. And this is the separase coming in and cleaving them. And now you can see them physically distributed. And then they end up in the two new daughter cells that are formed. And the chromosomes then begin to decondense at the end of that movie to form the two new nuclei of those daughter cells. And we can begin the process all over again. Right? I've watched this movie, honestly, probably 5,000 times, and I never get tired of looking at it. It's just, I don't know, just gorgeous to me, and just the precision by which it happens. You can see this is a slightly different way of looking at it. This is DNA stain, and this is a field of cells. And you can just watch all of these cells. We've got to make trillions of cells for our bodies. And so here in a dish, they're replicating and dividing. Mostly they're sitting in interphase, but every once in a while one triggers and it enters mitosis. Chromosomes quickly align, and then they separate to make those two new cells during anaphase. And then finally, I want to sort of zoom this forward a little bit. So the Walther Fleming drawing was good, but we can add color to it. So this is actually a picture I took a few years ago with the DNA in blue, the microtubule polymers in green, and the kinetochore structure on each chromatid in red. And so each pair of sister chromatids has to have two of these dots. And if you look at those, you can actually see the microtubules reaching out and sort of contacting those dot-like points. But we're going to zoom in a little bit on this. And so we're going to take and look at what a picture of a mitotic chromosome looks like. So this really is a chromosome spread from a cell. It's sort of been opened and the DNA has been splayed out. And you can see that the two arms are separated from each other, but there's a central contact point where they're held together. And this is where the cohesion is retained. The red dots are the kinetochores. You can see it's the big structure that's being able to form there. And that green is the sort of DNA that's sort of connecting them, still held together by those cohesin molecules. But actually, we can even zoom in further on this. And this is an electron micrograph of a mitotic chromosome. The dark stuff there, the electron-dense material, that is the DNA. Those lines that are coming in from the left and the right are the microtubule polymers. And then that big structure, sort of two big dark staining lines, that is the kinetochore. Okay? And so that's that contact point between those microtubules and the chromosomes. It's really a big machine that allows you to do that. And I really like this idea of machines. And so let's think about other machines in the cell. Ribosome is a great machine. It's a protein translation machine. Its job is to be able to make new proteins. Kinetochore actually is a lot like a machine in the same way the ribosome is. Its job is a chromosome movement machine. And so it needs to be able to hold onto these microtubules and distribute them. It's also a massive machine. So that's the ribosome down on the same picture there. And in this case, you can see that a ribosome, which we really think of as being huge, is just a small speck on what a kinetochore looks like. We're going to zoom in one last time and sort of take out what this is and put it into molecular parts. The microtubule has to touch onto this kinetochore. There's a second region which touches the DNA. And in fact, this really is a large machine where there's a lot of different proteins that connect that. So we have to have a microtubule, we have to have the DNA, and all of those other factors are parts of this larger machine. And in fact, there's more than 110 different players involved in this, and typically on the order of about 200 to 400 copies of each of them. And so this is much more complex than I can possibly diagram here, but we can bring these together to really see what this structure looks like. And so just in looking at this picture, I want to highlight just one major thing, which is that NDC80 complex. It's that long rod that reaches out to touch the microtubule. It's not the only thing that touches the microtubule, but it really is a key component of that interface. You need to build a larger assembly, and so there's a lot of things that recruit that complex there, for example. But that far point, those little dots that are holding on to the microtubule there, that is where Aurora B can phosphorylate. It can turn on and off that interface to allow you to bind or unbind to microtubules.